move on to lecture objective 2, which is describing the physical and structural support of the nervous system. All right, so this is again going to tie into a lot of what we spoke about in the anatomy lab. Right, the physical support of the nervous system is really the skull. Okay, that's the main or the basic support of the brain. We spoke about how the brain is very delicate tissue, very mushy, I think is the best way that I can describe it. Very sensitive, squishy. And so having the cranial cavity surrounding the brain is a protective measure from external trauma. Now, there's also the vertebral column, which protects or houses the spinal cord as well. Within the bone, in the skull, we also have the meningeal layers. And the meningeal layers are, again, helping to protect the brain, the sensitive, delicate brain tissue, from that very rough, hard bone of the cranial cavity. And so floating around within the meninges is that central spinal fluid, which helps to protect the brain as well. It's suspended in that fluid. The three layers of the meninges, we've talked about this, are the dura mater, the arachnoid matter, and the pia mater. And again, these are Latin translations to tough matter, meaning the outermost, toughest layer. The arachnoid matter, meaning the spidery layer. Again, that's a more functional sort of network appearing layer. It's not very distinct the way the dura mater is. And then finally, the pia mater, which is the deepest layer, and is very intimately connected with the brain and spinal cord, so much so that they are very inseparable. It's hard to differentiate or distinguish pia mater from brain or spinal cord. Okay? In fact, if you see the brain or you see the spinal cord, it is assumed or understood that there's pia mater surrounding it. Okay? That's your tender mother. All right. So this image really just illustrates those layers. So what we're seeing here is the skull. So you have the skin, which forms the scalp. And the scalp is really a mnemonic, actually, for the layers that form the skin around the skull. So there's S for skin, C for connective tissue, A is aponeurosis, loose areolar tissue, and then you have the periosteum, which is that first layer of bone here. Deep to the bone, then we get into the meningeal layer. So these are shown in blue here. All right, and the first layer is the thicker, tougher dura mater. Then we have the arachnoid mater here in purple. And again, this is not as distinct as this illustration shows it. Okay, it's very spidery and it's sort of connecting to both the dura and the pia below. And finally, the deepest, most intimately connected to the brain is the pia mater. Okay, so from deep to superficial. Now, it's important to understand that between the arachnoid mater and the pia mater, this is where the CSF lives. This is where the CSF is suspended and is circulated. So this is called the subarachnoid space, meaning beneath the arachnoid mater. Okay, and that is continued right around the spinal cord as well. So there's a subarachnoid space in the vertebral column, and that's where we do a spinal tap. Right, so if you go to the doctor, let's say you have some meningeal symptoms, they're thinking about meningococcal meningitis, or any of those bacteria that could affect the brain, they're immediately going to send you for a spinal tap. They're going to put a needle into your vertebral column. It's going to pierce the dura mater. It's going to pierce the arachnoid mater. It's going to be in the subarachnoid space, and then they're going to withdraw that fluid from that space and send it for testing and all that stuff. Alrighty, and this is a cross-sectional view again of those meningeal layers. Dura being most superficial, pia being most deep. Okay, and again, this goes right around the brain and right around the spinal cord. Any questions on that? Any questions? Okay, so CSF, we're not going to talk too much about the consistency of CSF. It's a clear, watery fluid. It bathes the brain and the spinal cord. It's similar in its makeup to plasma, but there are a few differences we'll talk about. And again, it's floating in those ventricles. If you remember the ventricular system, it should be a review from your lab topics, but it's floating through those ventricles around the brain epidermal cells of the choroid plexus, this we mentioned already, circulates through the ventricles, lateral ventricles, third ventricle, fourth ventricle. It ends up in the subarachnoid space where it's circulating around the brain and spinal cord, and then it is reabsorbed by the arachnoid villi. The arachnoid villi are these small openings that lead into the venous system. So the CSF that is circulated doesn't keep circulating infinitely. It gets reabsorbed back into the venous system, into the blood again, and then new CSF is made, and that process is cyclical and continues to generate new CSF, reabsorb, generate new CSF, reabsorb, and so on. Functions: to cushion the brain and to maintain a stable interstitial fluid environment. Okay, so it stabilizes the fluid that really bathes the central nervous system, as well as cushioning the brain tissue. All right, so this table sort of summarizes some of the differences between plasma and CSF. The takeaway point here is sodium, potassium, chloride, and calcium all have very similar levels in both plasma and CSF. Where we see some significant differences are glucose and proteins. So there's significantly less glucose and proteins in the CSF as compared to the plasma. So you don't have to memorize these numbers, just understanding that trend, that there's a lot less glucose and a lot less protein in CSF as compared to plasma. Levels of other ions like sodium, potassium, and so on are quite similar between the two. Okay. All right, any questions? Any questions? Okay, so this is a review here. I'm not going to go into much detail. We did this in the lab. The circulation or the flow of CSF from the lateral ventricles through the interventricular foramen into the third ventricle, which is really the surrounding area of the thalamus. It's going to go through the cerebral aqueduct and down into the fourth ventricle, and then from there it goes into the subarachnoid space around the brain and spinal cord, and then gets reabsorbed by the arachnoid villi. Okay. These arachnoid villi are found in various spots throughout the brain, and they're connected to the sinuses, which directly lead to the blood. Venous blood is going back to the heart. All right. Any questions on those structures, the ventricular system, the meninges? That should all be reviewed. All right, let's move on. Our third objective here is to describe the nature and significance of the blood-brain barrier. Here, when we speak about the blood-brain barrier, it's important to understand that this is a very important structure in maintaining the integrity of the nervous tissue. You don't want everything that's in your blood to be exposed to your brain tissue. Now, the nervous system accounts for quite a bit of our total body weight, about 2%. Okay. It's just the brain, spinal cord, and nerves. It receives a lot of your blood supply, so 50% of blood that's pumped out to the heart goes directly to the brain, spinal cord. Okay. And this is because the brain has quite a high metabolic rate. And when I say high metabolic rate, I mean that it's always active, it's always going, even while you sleep, while you sit, while you're resting, standing, walking, it's always active. So it uses about 20% of oxygen and about 50% of the glucose that is consumed just at rest. Not while you're taking an exam or studying or thinking, just at rest. We're using half of the glucose that we bring in, and we're using 20% of the oxygen that we bring in, just in the brain alone. So that's quite a bit. Okay, now the nervous system depends heavily on blood flow for energy. We'll talk about why that is. Oh, right here. All right, so the nervous system depends on aerobic glycolysis. And aerobic glycolysis needs what? What molecule does aerobic glycolysis require? Oxygen, perfect. So if we have no oxygen, we have no energy. This is why the brain is so susceptible, so sensitive to damage or injury if we lose oxygenation. The brain cannot make energy, it cannot conduct respiration or cellular respiration in the absence of oxygen, not easily. Okay? So it requires a steady supply of glucose and a steady supply of oxygen. Now there's not much glycogen storage happening in the brain, with the exception of the glia. We talked about this earlier. The glia, to a very small extent, can store away some glycogen that can be tapped into and use that for energy, but not most. That's not the majority of the time. That's not the major case. It's a very small extent. The brain cannot use fatty acids for energy. Right? We talked about how fatty acids provide more energy per molecule than glucose does, but the brain cannot tap into that fatty acid oxidation for energy. It is reliant exclusively on glucose. And so the brain needs lots of glucose because it doesn't get that fatty acid, which you can use to get more energy. It cannot tap into those stores. All right, ketones are used during extreme conditions. So ketones are those breakdown products of fatty acids. And so in very extreme conditions like starvation or diabetics, can the brain use glucose? So if you're on a ketonic diet where you're avoiding glucose and avoiding carbohydrates, your brain can tap into these ketones, but that's not a very efficient way to supply the brain energy. Really prefers glucose. Okay? Let me talk about strokes. This is caused by any interruption in the blood supply to the brain. So there are different types of strokes. We have occlusion of certain vessels, and we have hemorrhage in vessels. Right? So if we block a vessel that's supplying the brain, that brain tissue area will not get any oxygenation. It will die very quickly. It takes about a few seconds for the brain tissue to die or be irreparably damaged with deoxygenation. And the second type of stroke is a hemorrhagic stroke, which is basically a bleed. You have a tear in a vessel and you're bleeding out of that vessel. So you have an occlusive stroke and a hemorrhagic stroke, which both affect nervous function because they deprive the brain of oxygen. Alrighty, any questions on the blood supply to the brain? Questions?
The occlusion is blocks the vessel supply in the brain, and then.
Right, so if you block up vessels supplying brain tissue, that part of the brain tissue dies. So that's one type of stroke, an occlusive stroke. Let's say it blocks my PCA, my posterior cerebral artery, my entire posterior cerebral cortex loses blood supply, and that's a stroke. Very 5 to 6 minutes, very brief time, very brief time. And then the hemorrhagic stroke was different in the fact that you actually had a tear in that vessel. So you can obstruct the flow by just blocking the vessel, or you can actually have a rupture in that vessel where you have bleeding. That becomes a little more complicated. That blood actually constricts the brain. Alrighty, moving on to the blood-brain barrier. The blood-brain barrier, as I mentioned earlier, is that barrier between the external environment relative to the nervous system, so what's happening in your blood vessels, in the rest of your body, and the nervous system. It's important to have this barrier to prevent the brain from being exposed to all of the toxins, chemicals, medications that we are exposed to in our bloodstream. You don't want all of that going directly to the brain. The blood-brain barrier is important in maintaining that distinction. It's formed by capillaries in the brain. So it's formed by those capillaries, which are the site of exchange between the blood and the interstitial fluid. And then there's a thin layer of endothelial cells. Right? A thin layer of endothelial cells actually line this blood vessel. These endothelial cells sort of link together hand in hand by tight junctions. We talked about tight junctions already. These are those junctions where two cells hold onto each other so tightly that nothing can get through them. Okay, so the tight junctions between the endothelial cells prevents certain substances from traversing that blood-brain barrier, right? Things can be diffused, such as hydrophobic substances, small substances like certain gases, certain amino acids can cross. Alcohol, the particle that can easily cross the blood-brain barrier and into our brain, can traverse that barrier, but other substances like proteins, glucose, cannot easily move across the membrane. Glucose is going to need a transporter, that group 4 transporter that we spoke about earlier, to bring it across the blood-brain barrier. So the special anatomy consists of the CNS capillaries that will limit exchange, and then the astrocytes. If you remember, we talked about the astrocytes. They have that star-shaped appearance where they have many processes sort of sticking out. And at the end of those processes, they have what we call foot processes. You think of them as like little fingers that are grabbing an endothelial cell. If you think of an endothelial cell and the foot process of an astrocyte, when it sort of grabs onto that endothelial cell, it creates a very tight barrier where certain things can cross and certain things cannot. This is essentially the blood-brain barrier, very important in maintaining the integrity of the nervous tissue and preventing exposure to toxins and other harmful chemicals. All right, so this is sort of what I was just trying to explain here. So you have your endothelial cell, right, that thin layer of cells that are very closely connected to each other via tight junctions. And then interacting with these cells are the foot processes. So this is what we're calling a foot process. This is your astrocyte. This is one of its processes. And at the end of that process is this projection that's like a little finger that can grab. We call it a foot process. And that very closely adheres to these endothelial cells. And so in order for something to go from the blood to the brain, it has to go through this endothelial cell and through this foot process. And so these two structures together ensure the integrity of the brain and prevent certain things from going from the blood, right? Things that we bring in or are exposed to cannot just easily cross and end up in the brain. All right, that's your blood-brain barrier, and it's really formed by the tight junctions and the connection between the astrocyte foot processes and the endothelial cell. Any questions on this? Any questions? And again, this is sort of illustrating that large molecules like proteins will not traverse this membrane, right? Certain molecules would need a carrier, like glucose. So this would be a glucose carrier transmitting glucose from the blood out into the nervous tissue. And then some solutes that are hydrophobic will go right through the membrane, like alcohol, certain gases, amino acids, and so on and so forth.